All right, hello and welcome back to our PML School. Today we are presenting a highly popular topic, models for tumor growth inhibition. And again, we have as a reference an exercise from the 5th edition of the Gabrielsson and Dr book, and this is PD 43. If you have comments or questions, please post them into the corresponding panel on the right-hand side of the Cisco WebEx console. So let's start here. Here's an overview of today's presentation, and why do we need an overview? Main reason, there's one announcement at the end of the presentation that I would like to raise your awareness to, an extension of our PML library. We have translated a number of standard non-MEM models into PMLs, and these can now be downloaded from our forum. We will constantly add new models to that library, and maybe that's for the announcement. And apart from that, you see, today we are talking about tumor growth modeling, and as I mentioned in the invite, we have some general comments about this, a mini lecture. And then for the modeling part, we focus on preclinical models. So we've got Xenografts, modeling strategy, and we will present 3 different tumor growth kill models, Gompertz, Simeoni, and Jumbe. This is typical. I mean, tumor growth inhibition modeling is sometimes could be tricky, and you always want to test more than just one model. And that's why I've added a couple of more tumor growth inhibition models to the Jumbe one, which is presented in the book. Okay, so starting with tumor growth in general. Here's a typical profile. This is a curve which represents unperturbed tumor growth, so there's no drug treatment. And as you can see, it follows a sigmoidal shape. And some of you might remember our last session where Chris Mehl was presenting a bunch of different PD models to fit to a sigmoidal shape. And modeling tumor growth, like you see here, it might be much the same task. But it's, and also Chris Mehl did present the Gompertz function as one of those models that he presented, and this is actually a classical function that is used in oncology. And why is that? You see that in the early phases. Let me show you. Here's that pointer. In the early phases, we see exponential growth, and then this tumor. Then with time, as the tumor size increases, the growth will decrease until it eventually stops. What happens behind the scene? In order for the tumor to grow, it requires nutrients, oxygen, and space. And for the transport of these vital resources, the tumor requires a vascular system. However, the vascularization cannot keep pace with the tumor growth. And that is why tumor cells go into a resting mode or gene zero phase, or they eventually die. And in effect, what we are seeing is that the tumor growth decreases until the tumor has reached its maximum size, which is shown on top here. Also looking more closely at the curve, you can see that this curve is not symmetric to its inflection point. So the inflection point lies a little bit below the symmetrical in the middle of the curve. And here, that's why the Gompertz curve is able to capture this behavior. And this is why it's also been widely used in oncology in describing tumor growth. So this is a Gompertz function with its 3 parameters, the TV0, the initial tumor volume, initial growth rate constant, and deceleration rate constant. But if we want to study cancer or tumor treatments, there are 2 distinct thresholds. The one at the bottom, this is a detection level. So in the beginning, the tumor is growing unnoticed, and when it hits this threshold, this is a detection level, it gets diagnosed, and from this moment on, the clinical phase starts. But the tumor continues to grow until it hits a second threshold. And this is when the tumor was getting too big and is interfering with vital body functions. And then, as a result, the patient dies. Move on. The time span between those, of the curve between those thresholds is known as survival time. The primary goal in cancer treatment is to increase the survival of patients, or in terms of modeling, to expand the time to such an event. This can be achieved by decelerating the growth, or inhibiting the growth, or even stopping the growth, and later on eradicate the tumor. Okay, but this can be a very, just a second. This expansion, I mean, this can be a challenging task because you have to consider adverse effects and also biomarkers. In addition to this tumor response model, which is all driven by the PK model that is describing the concentration of the drug in the body. So adverse effects are important since most of the cancer treatments have a very narrow therapeutic index. And this makes sense since the drugs are toxic and are supposed to kill cells, preferably tumor cells, but in most cases they do hit normal cells as well. Also, the tumor size itself is not sufficient to describe the complexities of cancer. Biomarkers are needed to monitor various aspects of the disease and its treatment. I'm showing this slide with this complex network of models just as an example of advanced PK/PD modeling. This complex framework of interdependent models is beyond us right now. But at the same time, I know we all have aspirations, and some of, or perhaps many of you may want to reach this stage. I can only encourage you and say, make it yours. With the basic knowledge that you've gained in this PML school, you have a very good foundation to build up on, and step by step you will learn how to manage to deal with more complex models. But maybe stop dreaming for now. Let's get back to our level at the beginners or maybe intermediate stage. In our session, we want to focus on the tumor response PK/PD model. This task is tricky enough to be of interest, but at the same time, it is simple enough to demonstrate how to implement the code of models in Phoenix Bernoulli. So we are leaving the clinical scenario and focus on the preclinical or drug discovery scenario. Here we are using animal models to study cancer. Xenograft models are the most common in vivo models in oncology drug development. In drug discovery, you might have identified cytotoxic compounds, but you don't know exactly what individual targets, biochemical targets the compound hits. In this situation, xenograft models are often used to find out if a compound is active on tumor cells or not. So it's really there to identify active compounds. And for xenograft models, you take human tumor cells and implant it under the skin of nude mice. Then there is a solid tumor that is developed, and once the tumors have reached a certain size, the mice are randomized into different treatment groups. There's one group with, without treatment, without drug treatment. That is a placebo group to study the unperturbed tumor growth. And typically, you also have another control where the mice are treated with a drug that is known to be active on those tumor cells. Then, according to the protocol, the doses are given and the tumor size is measured over a period of time. This measurement is simply done with a caliper where you measure the length and the width of the solid tumor, and using some equations you can estimate the tumor volume. And this tumor volume is the effect or response data that we take for tumor growth modeling. Before we start modeling, just a few comments on modeling strategy. First of all, the PK. On the pharmacokinetic side, we do not have a lot of PK measurements done during a xenograft study. Just a few satellite PK measurements, maybe on the first and perhaps the last day of the study. For PK/PD modeling, you assume that you have characterized the PK of the compound beforehand. So you can freeze all PK parameters for the PK/PD model. Then modeling the tumor dynamics is done in two stages. First, you model the unperturbed growth to find out about the aggressiveness of the tumor and also the resistance of the animals. Then in the second stage, you model the drug-induced decay of the tumor growth. Here, the ultimate goal is to rank order the compounds based on their potency against the tumor. Since we are dealing with rather complex models in these scenarios, it is advised to run a sensitivity analysis on the individual PK/PD parameters. You will eventually find out that some parameters are not sensitive at all. But finally, when you are done and you are convinced that you found the right parameter estimates, you can use your model to do simulations. And you can simulate new dose levels or new dose regimens. Or eventually, you can also use your model to do allometric scaling to identify the first dose in human for phase 1 studies. So this is the end of my little mini lecture on tumor growth modeling. Now starting with a typical session. And here we go to the exercise PD43 from the Gabrielsson and Dr. book. And the objective here is to characterize tumor growth kill using 3 different models. We start with a Gompertz model. We've already seen the function. And we then move to a very popular one, the Simeoni model. And finally, take on the Jumbe model, which is actually the exercise that is from the book. The goal here is to find estimates for the tumor growth rate and for the tumor kill rate. So let's have a look at the data. Here's the plot, the linear plot of tumor volume versus time. In our xenograft study, we have 4 different treatment groups. The yellow one here is a placebo one, and then 3 groups with drug treatment at escalating doses, 2 with rather low doses, and 1 with a very high dose. And you can see already, when this is the unperturbed growth curve, due to the drug treatment, the growth gets inhibited. And here, with a high dose treatment, you can also see the tumor eradication. So at first, after the compound disappeared from plasma, then the tumor size increases once again. So for this data set, we want to find good models. Let's see. And as usual, we are building our model in a stepwise manner. We're starting with the built-in options. Here we leave the population box checked because we have 4 different treatment groups, and we want to find parameters that fit to all 4 different treatment groups. We switch the type of model to PK linear. This is the most simple PK/PD model. For the parameterization, this time we switch from clearance to micro, just because we are sticking to more closely to the exercise in the book. Then we change the absorption to extravascular. And here we check the T-lag box as well as the freeze PK box. So that leaves just the PD parameters for the fit. So that's all we need from the built-in options. We have already half of the code ready for our final model. So we go to as usual, to the graphical model builder. Here we don't apply any changes. And this is then switch over to the textual model. Here's the PML code for this, for the built-in options. The only thing we need to do here is to change the effect equation, which is here just E equals alpha, and this has to be replaced with a Gompertz function. The rest stays the same. Again, what we need to do as well is we will delete those ST-PARM or structural parameter statements. We just stick with the fixed effects statements. That's what we do in the next slide. So here we have the different differential equations for the Gompertz model. This is how it looks like. And for the decay, we have a proportionality factor gamma that we use here. And we can combine the Gompertz function with the decay function in this manner, just like this. And this is actually exactly the code that we need to write into our model box. And you see this highlighted here. It's exactly the same. So, okay, as I said, we change a little bit the code. We need to enter the sequence statement, so to initialize the tumor growth. The first step is the effect, which is a tumor volume equals the initial tumor volume, E0. We've got the PK parameters frozen. This is coming from the PK model, and then we've got a few fixed effects for the PD model, for the tumor growth inhibition model. The initial tumor volume, A, the growth rate constant, B, the deceleration rate constant, and gamma, or G, as the drug decay rate constant. So that's the Gompertz model. Now switch to the Simeoni model. Before going into the code, actually, just a few comments. The Gompertz model is known as an empirical model. It is only describing tumor growth. And when you change the dose level or the regimen, you will expect also some parameter changes. To the contrary of this, and there's a mechanistic model, which gives more a sense of biology behind the tumor volume and the growth. So this considers typically different cell populations, cell cycle kinetics, and also biochemical processes. But in order to model this, I mean, this requires a lot of assumptions, and these assumptions need to be modeled with parameters, and either you have already found them, or you need some experimental observations to get good estimates for those parameters. And this is typically very costly in terms of time and effort. So there's a Simeoni model, which is a compromise. It's a semi-mechanistic model. It is based on less assumptions and requires only typical preclinical data, such as the PK model, so PK parameters, and tumor response data. So there's nothing extra for this model. And we will get to this now. So this is a schema of the Simeoni model. The major assumption here is that there are different cell populations, E1 to E4. And we have 4 compartments. These describe the different cell populations. In E1, these are the cells that are growing. So these are the proliferating or cycling cells. And they run through the typical processes. And this compartment describes the unperturbed growth of the tumor. And it is assumed that the drug is acting just on these proliferating cells, not on the other ones. So this is the equation. It's also proportionality factor K2 times the concentration times the population in E1 of the cycling cells. Another assumption of the Simeoni model is that there are 2 growth phases. Initially, there's an exponential growth phase. This is expressed with the parameter lambda 0. You see lambda 0 times E1. And but then after a certain time, the growth becomes more linear. So in this case, and for the linear phase, there's another 0 order rate constant, which is lambda 1. And this assumes that you will never reach a sigmoidal curve. You will never reach the plateau of the sigmoidal curve. And this also makes sense is because in typical xenograft studies you will never reach a plateau. And the animals are also sacrificed before they reach too large tumor sizes. So in this case, this is a complex construct, a mathematical construct here, which just it describes a switch between these two different phases of the curve. And so I don't want to go into details, but it's such that depending on the E1, on the size, the tumor volume, the size of the tumor, in the initial phases, it is dominated by the nominator term. But once it reaches a threshold volume, it switches over into the linear growth phase, and that is being achieved by this construct with a high exponent. This is psi, and this is typically set to a high value like 20, in order to provide a sharp switch. And then you have this drug-induced decay, which is just described by a proportionality factor K2. So this is mainly the description of the first compartment, everything what happens on the proliferating cells, and once they get killed by, or not killed, damaged by the drug action, the cells move into these different transit compartments. And the transit compartment represent different stages of the tumor cells on their way to the cell death. And transit compartments is a typical way in modeling when you have a distributed delay. And in this particular case, it's a delay in cell death. So the flow between those transit compartments is described by a transit rate constant K1, and this is assumed to be the same between all transit compartments. So here you see the equations for the transit compartments E2, E3, and E4, where E2 is the input from E1. This is a drug-induced decay, all the population, all the cells that are killed or damaged by the drug, minus obviously the flow out of the E2 compartment into E3 and so forth. And the total tumor volume is then just the sum of the tumor cells from each compartment. So that's the model. Let's see how we can put that into the PML. So this is, I mean, it's if you remember, these are the equations, right? These are the differential equations, and what we do, I mean, it's just writing exactly those formulas into the PML. So you see, it's mainly the same code. I mean, mathematical code, and you see it here. And I've written the denominator part of the E1 compartment just in a separate line in order to make reading more easy. And again, with the Gompertz, we had this before. I mean, we have to initialize the tumor volume, so E1 equals E0. So at the start, there's an initial tumor volume. And that's the rest is much the same code as we saw from the Gompertz model. There are a few secondary parameters that I've coded here, like the threshold tumor volume. This is the tumor volume when it switches from exponential growth to linear growth. We also have a threshold concentration for tumor eradication. This is also very helpful because this concentration is useful to predict new dose levels or new regimens, where the goal is to stay above this threshold concentration in order to really eradicate the tumor. Now this was the Simeoni model. Let's switch to the Jumbe model. This is actually the one from the book. So this is much the same as the Simeoni model, as you see, and it distinguishes between those two different cell populations. And for the distributed delay, Jumbe also uses 3 compartments, 3 transit compartments, but there are 2 major differences. First of all, the growth, it only assumes an exponential growth of the tumor. And it is described by this first order rate constant K growth. It uses, for all the compartments, a power term 2/3. And this is on all compartments, the cycling cells as well as the dying cells. Also different is the drug-induced decay. It's using a saturable drug action, and it is expressed with an Emax model with K-kill-max and KC50 as parameters. But apart from these differences, everything looks much the same. So if you go into the code, you see, okay, it's similar to the Simeoni model. We just need to change the inhibitory function, which is here, this first line. And also the transit compartment needs the power terms, right? And there are slightly different naming conventions for the parameters. Also, you see here some new statements like E11 equals max, E1 or 0. I mean, this is, I guess, related because Dan Weiner wrote the code and he doesn't like misbehavior. So whenever the effect becomes negative, a value of 0 will be used instead. Okay, but that's the model. Okay, the only thing that we need before doing the demo is finding initial estimates. And it is quite difficult to find new estimates with these complex equations. That is why I prefer to use the initial estimate step of the Phoenix model object. An alternative would be to use the profile run option on Phoenix. For this, I mean, if you've got the population mode checked on, it's a different run mode, profile. And here you can activate, you can check some of those parameters for which you want to do a sensitivity analysis, and then you have to provide some perturbations here, and for example, in form of percentages of the set value. And what you get out of this is a plot of log likelihoods over the parameter value range, and where you're looking for a maximum, and this should be the ideal starting point for fitting. Okay, so I guess this is now time for the demo. Okay, let me share my screen. Okay, so what I've got here is, let me just see, this is the data. So we've got some dose, a small dose table, and the PD43 data, time, tumor volume, and we've got 4 different treatment groups. So first let's plot the data. We send it to a XY plot object, just get here. And time to the X, tumor volume to the Y, and group by dose group. And this is a plot. This is what we've seen in the slides. So starting with the Gompertz model, we will send this data set to a Phoenix model object. Okay, we just call it accordingly Gompertz. And as I said, for the built-in options, we stick to population. We change the type to PK linear. And as I said, because we want to be close to the book, so we change from clearance to microparameterization. Absorption is extravascular. We wanted to deal with differential equations, so we just check up of the closed form, check the T-lag, and then freeze the PK. Okay. Now we need to do our mapping. So dose group, as I said, we want to use the dose group as an ID, and tumor volume is our effect observed. Now let's quickly have the doses defined here. Again, dose group is ID, time is time, and amount is AA. Okay. With that, we've got everything set up. We can go to graphical mode. So it looks like this. Typical one compartment extravascular model with an PD block and an additional parameter TLAC. But we don't change anything here, so we go directly into model as textual. So we've got this. This is the model. The first one is as we've seen. Wait a second. It's not. Let me go back. I think we made one. Why aren't the PK freeze PK? Yes, added as textual. Yeah, here now they are frozen. The PK parameters need to be frozen, and just the PD parameters are there for fitting. But obviously we need to change this one, and we need to add some PD models as well. So, but I mean, I can write down the equations here like they are, but I guess to save some time, I'll just take this and copy it from the textual model. And this is exactly the same. We provide these model text files after the event in our forum. So this is the same thing you can do when you want to reproduce this session. There's a model. So we just take away this one until that and just copy it in here. And no warnings, so we just can go to the initial estimates tab. And see whether we need to change some of the settings. Initial estimates. We need a duration of 800 hours. And we need the overlay. So let's see. So we've got, these are the for PD parameters, E0, A, B, G. So E0 is 350. That's about right. A is 0.05. The G function could be a little bit larger, maybe. Yeah. Something like this. Maybe we can decrease B a bit. This is something you can. You see, this is how you can get closer. Too quick. It doesn't need to be perfect, right? So something along this one. Something along these lines. Okay, when we check this, 350, 0.05, 0.06. We need to change B. Maybe we make it a little bit smaller as a starting point, and we need to increase G to maybe 0.6. And with these initial estimates, we can start and do the fit. Okay. Doesn't want like me. Oh, wait a second. We have to change the method to naive pooled. Sorry, forgot about that. Let me do that once again. And this option is available with the Phoenix Vinodin license. So now we got the fit. Let's have a look. Oops. Come on. Here you see the fit. It actually matches those first curves quite well. It has some problems with a high treatment group. What it does here doesn't look too great. So maybe we find a better model. So let's move on to the Simeoni model. So we copy just our Gompertz model, paste it back, and call this Simeoni. And here again, I guess we could just copy and paste from the text, because otherwise I would be writing here for minutes to come. So let me do this one. Here's the Simeoni model. We just need the PD part. Come on. Copy this and paste it back here. Okay. So this is the code. Let's also look here at the initial estimates. How are we doing here with first parameter? This looks actually quite good. You see, I mean, we can change the exponential growth and the linear growth. Quite sensitive. And you can change the transit rate constant and the drug-induced decay constant. But these look actually quite good. Just compare 0.05. It's okay. 3 is okay. 340 for E0. And it is okay as well. So we can just run this, execute here. And look at the results. Let's see. Okay. This you see here. I mean, the low dose treatments are very nice fit, and also the high dose treatment. You see a good fit here as well. So this is much. It is better as a Gompertz model, I would say. Yeah. So let's move to the next one. Copy. Paste again, and this will become the Jumbe. Same procedure, just replacing the PD part of the model. And here's the textual model. Let's see. Come on. Ah, come on. So once again, copy and paste here. Okay, and here again we look for the initial estimates. Okay, this looks not great. Let's see. Maybe we can increase the growth curve a bit. And maybe a little bit kill. Oh, the KC50 is way too high. Let me see if I can get this down. This one is better. Maybe something like this. Okay, grows. Okay, it should be good enough. Good enough. So let's see. We need to K growth is 0.03. Let's change this in the code as of initial estimates. This one is 0.1. It is good. K max is maybe 14. And this one needs to be 1. And 350 is okay. So with this one, with these initial estimates, we can go. And it takes a bit longer. Let's have a look at the plot. And let's see. Okay, here you see it really nicely fits those placebo and low dose curves as well as the high treatment. But it's maybe a little bit better than the Simeoni, but not too much. In order to judge that, I mean, we can insert maybe a comparer object, a model comparer object. And here we can, these are the three models that we want to compare. We don't have random effects, so we can switch off these tables for comparison. But we want to look at the individual plots and just run it. Come on. These three, okay. Execute? No. Okay, it doesn't like, I mean, there are some names come up here quite often, and so, okay, but these values are pretty close or even identical when you look at this. So let's have a look at the overall table. We just sort maybe by Ikaike criterion. And here you see that the Jumbe model is the best one in terms of Ikaike, but also in terms of PIC. Simeoni follows after that, and the Gompertz is the worst in this series. If these 10 points are statistically significant, I can't tell right now because it needs some chi-square test to do that. But let's have a look at the plot. Here you see the different plots, and you see that the Jumbe is really very nice. The Simeoni is not bad, but I guess the Jumbe is dealing better with this high treatment group, and with the Gompertz, this can't really fit the high treatment group properly. Okay, with that, I guess I'm done with the demo. Let's go back to the slides. Just a summary. The results, as we've already seen, the fits for the three different models, and also the diagnostics, the Ikaike and Bayesian criterion. This reflects that the Jumbe model seems to be the best choice for this particular data set. What we can do, but maybe it doesn't make too much sense, is to briefly compare the numerical outcomes, the parameter estimates. But apart from the initial tumor volume and the residual error, those values are difficult to compare one another because you see different. If you're looking for the exponential growth rate constant, which is present in all three models, these values differ. There is also a drug-induced decay rate constant in each of those models, but also here the values differ. And that is expected since the differential equation used to get those estimates are different. And you can use this for ranking. I mean, if you have more compounds to compare the ranking, I mean, you could use those values. Finally, just a summary on the typical aspects of the different tumor growth kill models that we've used. So we started with the Gompertz model, which is a classic equation used in oncology. It is an empirical model where the tumor growth is expressed with first-order rate constant. And this model assumes the drug effect to be applied to the total volume, total tumor volume, and where the drug-induced decay is expressed by a proportional factor. The Simeoni model is a very widely used model in the preclinical stage. It's semi-mechanistic, so it differentiates between two different growth phases, exponential and linear. It also differentiates proliferating cells from dying ones. and drug-induced decay is also expressed by a proportional factor. Finally, the Jumbe model is also semi-mechanistic, similar to Simeoni, because it distinguishes between the two different cell populations. It expresses the growth phase with a single exponential growth, and it uses a separable function, an Emax model, for the drug-induced decay. So with this, I guess I'm at the end of my presentation, and I'm coming now to the questions. I mean, we are already a little bit behind in time, so maybe just a few. So we start here with can transit compartment be added in Gompertz model? As we said, I mean, let's go back to the equation here. So it has a single growth exponential function, and you know, this is the same as with the Jumbe model, which uses, you know, transit compartments. Yes. So I mean, this is what the tumor growth inhibition models are all about. They do a lot of combinations, right? From where you have some parts like the growth phase or the kill phase or whatever deceleration phase, and the drug-induced decay. As you see, here's also a combination of 3 different themes, if you will, and these are mixed between these 3 models. And so you can, you know, use it like modules. You can replace one with the other and so forth. So that should be possible. In the Simeoni model, what is E versus E1? I think this is something I tried to explain. So let me go back. Here, E1, E is the total volume, right? So that is actually our measurement. This is our effect that is our response that we are modeling. This is a measured tumor, the predicted tumor volume. But we say the predicted tumor volume is a sum of all cell populations of the cycling cell population as well as the dying cells, right? So this is E versus E1, and E1 is just the source part which is proliferating where the drug can act upon. Because in the Gompertz model, just to see that, in the Gompertz model, the drug is acting on the total tumor volume, which is different from what Simeoni is doing. Here the drug is only affecting the cell population in E1. How can we get in? I guess for the rest of the questions, since we are already way beyond our time, we will get back to you and prepare responses. And as usually, we will upload them to our forum so that you can to make it available there, together with the slide deck and the link to the recorded session as well as the model text. So let me. Just as I said, I had some announcement to do. So this is it. As I mentioned at the beginning, I want to take this opportunity to introduce you to a new extension of our PML library. As you know, I mean, this is the 13th session of the PML School, and we've already uploaded about 20 different models that you can download from our forum. But we've expanded here with a new area of our forum, which we call NLME non-mem model comparisons. Here's the URL to find it, and this is just a screenshot from our forum. You see, we've already published the first models there, 12 different models, and let's go there in depth. If you go, I mean, these are all the different models that are published. So if you go into one link, you will see, this is, for example, the one with the PK2 compartmental oral with disease state covariate on volume and clearance. What you find there is a short description of the background of this model. And you have a slide deck, this one here, that goes into detail how to translate the non-mem control stream into PML code. This is described in the PDF. And then you've got a Phoenix project file. And the Phoenix project file includes, first of all, the the non-mem control stream as well as the PML code, but also executed models. So what is being done here is there's a non-mem shell script available in Phoenix that you can launch the non-mem run from within Phoenix and get back the non-mem results into Phoenix. So this is being done in this Phoenix project file, as well as a corresponding Phoenix model. This is also being run, and you can compare the differences, which there shouldn't be big differences between the two outcomes, between the two results. But you can compare it within this Phoenix project file. So we are working on further extending this library, so there should be eventually 40 to 60 different individual models in this part of the forum. But we are also curious about any feedbacks that you may want to give on this extension of the PML library. Before we close, what's coming up next, I would want to raise your awareness to this webinar. It's something we've touched upon in this session, distributed delays. We had this distributed delay of cell death, and we've used a transit compartment as a way to get it. But Wojciech Krzansky will present PKPD systems with distributed delays in a more general way, and this session goes live on May 16th at 11 a.m. Eastern. Not to forget the next PML School session, which is on April 27th, modeling inhibition of enzyme activity by means of turnover, and this will be presented by Chris. Before we close, just to make you aware, there are still some sessions to go until we end with our first series of PML School sessions. These are going until August 10, and we are already in the planning of an extension to this series. But also, would be good to hear from you any feedback, what you want to see. Maybe you can also send us feedback. Okay, these 20 sessions that we've presented, this covers everything. I don't need any more. It would be good to know. So any feedback that you have, please can you forward to me, and I'll be happy to respond to that. With that, I guess I'm at the end of this session. Thanks everyone for attending, and I hope to see you at our next session. And so goodbye, and you may want to disconnect now.